So hello students and welcome back again to another video tutorial by Genome Biotech. In this particular video tutorial, we would be talking about one of the most important topic for CSI net, the bacteriophage lambda, and how the bacteriophage lambda is being regulated, in what manner the regulation of bacteriophage lambda takes place. Okay, so what is this bacteriophage lambda and why it is so much of importance? Firstly, the bacteriophage lambda, as you can see, this is the bacteriophage lambda. It has its natural host as the E. coli bacterium. It has a natural host, and that natural host is the E. coli bacterium. It performs both lytic as well as lysogenic cycle. So this is a temperate phage. The phages are of two different types. Whenever you talk about phages, the phages are of two different types. Okay, whenever you talk about phages, the bacteriophages are can be a temperate phage, it can be a virulent phage. A temperate phage is the one which performs both lytic as well as lysogenic cycle. Temperate phage, and another one is referred to as the virulent phage. Temperate phage performs both lytic as well as lysogenic cycle. Both lytic as well as lysogenic cycle, as well as the lysogenic cycle. Lysogenic cycle. Okay. Whereas the virulent phage has an ability to perform lytic cycle only. It performs only lytic cycle. Okay. When you look at the things such as in the case of temperate phage, the example of the temperate phage include your lambda phage. M13 is a temperate phage. M13 phage. 5x174. 5x174 is a temperate phage. Virulent phage will be the phage that would be performing only the lytic cycle and never the lysogenic cycle. So we would be looking at how the bacteriophage lambda works, in what manner the bacteriophage lambda works, and what is this bacteriophage lambda. Okay, so let us start with this our topic. Now, transcription switches are there in the bacteriophage lambda that have the ability to switch on the transcription or to switch off the process of transcription in the case of bacteriophage lambda. So, looking at the cycles of the bacteriophage lambda, how the cycles of the bacteriophage lambda are being regulated. There are basically two different cycles that operate in any of the bacteriophages across the globe. The two different cycles that operates are the lytic cycle and the lysogenic cycle. Now, both the cycles operates in the globe, and one is called as the lytic and another one is called as the lysogenic cycle. So what is lytic cycle? Infection by phage produces many progeny and breaks open or lyse the host bacterium. So in this, the lysis of the host bacterium is going to take place. This is referred to as the lysis cycle or the lytic cycle. In the case of lysogenic cycle, what happens is that after infection, the phage DNA integrate into the host chromosomal genome and will live there passively. This will lead to the formation of a prophage molecule. Okay, so it will lead to the formation of a formation of a prophage. So prophage is important. So the prophage is going to be produced because this is the one where the prophage is going to be produced. Now, when the virulent bacteriophage enters into a cell, it only performs one type of cycle that is referred to as the lytic cycle. But when a temperate phage enters, it can perform both lytic as well as lysogenic cycle. Okay. So lytic cycle are being performed by the virulent phages, whereas the lysogenic as well as lytic cycles both can be performed by the temperate phage. These types of phages are called as temperate phages. These are called as temperate phages. Okay. Now, in the case of lysogeny, there is no progeny. No progeny is being produced. No lysis of the host bacterium takes place. That is, the bacteria is not being lysed. Can subsequently lyse because they can lead to the formation of lysogeny through lysogenic inversion. Now, there is a phenomena in which a lysogen can get converted into a lytic phage, and this phenomena for the interconversion of a lysogen into the lytic phage is referred to as lysogenic inversion. It is commonly referred to as lysogenic inversion. Lysogenic inversion. Okay, this is referred to as lysogenic inversion. This is lysogenic inversion. Bacteriophages lambda can do either. Okay, so they are the one which are the temperate bacteriophages. They can perform the lytic cycle. They can perform the lysogenic cycle also. Now moving forward. Now see, you can look here that, see, this is a phage that has entered its genomic DNA into a cell. When the phage has entered its genomic DNA into the cell, then it can cause the integration of its genomic DNA into the cell, called as lysogeny. If UV light induction or any mutagenic agent is being added, then in the presence of the mutagenic agent, the phage will undergo, in the presence of a mutagenic agent, the phage will undergo interconversion. It will be interconverted. How it will be interconverted? It will get converted into a mutant phage. It will get converted into a lytic phage. Okay. Instead of performing the lysogenic cycle, this phage, which was referred to as prophage, will get excised off. When this prophage will get excised off, this prophage will get excised off, will lead to the formation of lytic cycle. This phenomena is referred to as lysogenic inversion. Lysogenic inversion. Okay, already told to you. Now, there is a term which is being used. One is referred to as the episome, another one is referred to as the prophage. Now, when you talk about the two terms, episome and prophage, how the episome and the prophage are different from each other. Okay, so when you talk about a term that is referred to as episome, in the case of episome and prophage, the differences which are present. In the case of episome, the plasmid DNA has integrated into the bacterium DNA. When plasmid DNA gets incorporated into the bacterium DNA, when the plasmid DNA has incorporated into the bacterium DNA, has incorporated into the bacterium DNA, into the bacterial DNA, then it is referred to as the episome. When you talk about a prophages, when the phage DNA has entered into the bacterium DNA, into the bacterial DNA, when the phage DNA has entered into the bacterial DNA, has entered into the bacterial DNA, then it is referred to as a prophage. Okay, so this is the main difference between a prophage and an episome. This is an episome and this is referred to as a prophage. Okay, any doubt, you can just use the chat box to communicate with me and to ask your doubt. Moving forward. Now, elements of lysogeny. What is lysogeny? The phage genomic integrates into the host bacterium cell is referred to as prophage. Bacterium carrying the prophage is referred to as lysogen. The bacteria is referred to as lysogen. Lysogens are immune to further infection by similar phages because the phage function as repressed in trans. Now, I'm going to explain this particular process. Induction of the lysogeny leads to excision of the prophage, replication of the phage, and lysis of the host bacterium as the induction of the lysogeny takes place. In the case of induction of lysogeny, what is going to happen? In the case of induction of the lysogeny, the phage is going to be excised off. As it is going to be excised off, it is going to replicate. And then as it is going to be replicated, it is going to move forward. And then it will lyse the host bacterium. It will be causing the lysis of the bacterium to take place. And the bacteria of the host cell will be lysed. This is how the process of induction is going to take place. This is the manner in which induction is going to occur. Now, the next point that we have to start is about this particular point. Lysogens are immune to further infection by similar phages. Why the lysogens are immune to further infection by similar phages? And what is the strategy that a phage carries along with itself in order to select its host? Please look at the slide previously shown to you once more. Now see, phages always possess a strategic advantages within themselves. Phages, whichever are present, they have a particular strategy inculcated within their genome. Now, what is that strategy that we have to see? See, this is the genetic material of the phage. You look here. If I'm going to amplify this out, then what I'm going to show it to you. Now, see, this is the genetic material of the phage that is present. Okay, we are going to look at the genetic material of the phage. Now, in this genetic material, you will find the double-stranded DNA is there. Of course, the double-stranded DNA is there. Now, this double-stranded DNA, I have made the double-stranded DNA from in front of you, possess specific sites. Bacteria, to counter the attack of the phage, has made certain bacterio bacteria has made certain restriction endonucleases within itself. Endonucleases. Now, these restriction endonucleases are the enzymes that will lyse the double-stranded DNA molecule at a specific location, at a specific site. They are going to lyse the DNA molecule. Okay, but those specific sites, which would be the site for the action of bacterial restriction endonucleases, the virus develops its host range by protecting its DNA at these sites by catalyzing methylation at these positions. These positions are highly methylated so that the restriction endonuclease won't be able to cleave the bacteriophage genetic material because all these sites have been fully methylated. Okay, this is site for action of restriction endonuclease. Site for the action of restriction endonuclease. For the action of restriction endonuclease endonuclease enzyme. Okay, so all those sites have been methylated. Now, because the sites have been methylated, the bacterial restriction endonuclease won't be able to cut at these precise location. They will be inhibited. This is RE, restriction endonuclease of the bacteria. So restriction endonuclease of the bacteria, when are not going to degrade the DNA of the phage, then such type of phage will be included within the host range of an organism. And host range of an organism refers to the different types of organisms that the parasite could infect. This is Lambda Phage, and in its host range, it has a host range to infect E. coli. Okay, so E. coli could be infected. Okay, it could infect it because it has catalyzed methylation at all those locations where the restriction endonuclease of the E. coli is going to act. Okay, thus the DNA of the cell will be completely protected from degradation. Okay, now, but when this lambda phage will enter into the Staphylococcus aureus, for example, let's suppose, for example, what happens if lambda phage will enter into Staphylococcus aureus? If lambda phage enters into S aureus, if lambda phage is going to enter into the S aureus, then what is going to happen? Then the next phase of reactions are going to take place, and you must understand how the reactions are going to be proceeded now. In this case, the phage will have different set of restriction endonucleases. Those restriction endonucleases will catalyze the cleavage of the lambda phage genomic DNA. This is most important. Try to understand this point. If this is going to enter into the organism such as Staphylococcus aureus, then the genetic material of the lambda phage will not be able to protect itself from the Staphylococcus aureus, and it is going to die off. How? Look here. See, now the genetic material won't have that type of methylations that are being needed for its protection against the staph. Now see, I'm again making its genetic material just for the sake of understanding. Now, see, this is the double-stranded DNA. It has the pattern of methylation for protection against restriction endonuclease of lambda of E. coli, but it won't have those for staph, and so staph restriction endonuclease are going to cleave it. So it would be cleaved by staph restriction endonucleases. Okay, this will be cleaved by staph. S aureus RE. Thus, S aureus RE, thus S aureus is outside the restriction, is outside the host range of lambda phage of the lambda phage. So it is going to be outside the host range of the lambda phage. Lambda phage won't be able to cleave this particular double-stranded DNA because it is completely out of the host range of the lambda phage. This is how the host range of any virus is going to be decided. If the COVID-19 has been able to infect the human beings, then it is because the COVID-19 has altered its genomic DNA in order to enter the host range of the humans. In order to attach the humans into its host range, it has modified itself. That is why we are being able to being infected by the COVID-19, else not. Okay, next, moving forward, hopefully this would be clear to you. Next, now moving forward, just deleting the annotations. So COVID-19 has modified itself, and thus from being zoonotic, it has entered into the human beings because of the high rate of mutation persistent with COVID-19 virus. That is why it has altered its host range. Now moving forward. Now, elements of lysogeny. I have already told you, lysogens are immune to further infection by similar phages. Once the phage has infected a particular lysogen, then this phage makes either the receptor for its own entry to be degraded. The phage will synthesize certain proteins. This phage will synthesize proteins, and these proteins will act as proteases. These proteins will act as proteases and degrade the receptor. They will act upon the receptors that were responsible for the entry of the phage, and those receptors will be completely degraded. See, every phage require a receptor to be present within the bacterium so that the phage can enter into the bacterium. For the lambda phage, the receptor is TON B receptor. This is lambda phage. And for this lambda phage, there is a receptor that is present, and that receptor is called as TON B receptor. This is referred to as TON B receptor. This is the receptor through which it has entered into the bacterium. Once it has entered into the bacterium, it is going to integrate. Once integrated, it will form the prophage. Okay. And once it will be integrated, it will synthesize protein that will either degrade this TON B receptor or internalize this TON B receptor. Now, moving forward. Already told you this particular slide. Now, see, this is referred to as the segment of genes which are present in the case of Lambda Phage genome. You must understand that the genome of the Lambda Phage contains of more than 50 different types of genes. Do not get confused with all these different types of genes. I would be explaining to you all the different types of genes, how their interaction takes place, how their work occurs. Okay, the genes are C3 and C1 CRO and C2 genes. These are the genes that are responsible for the process of transcription to take place. The genes are C3 and C1 CRO and C2 genes. Okay, so these are the different types of genes that are being present within the lambda phage genomic DNA. There are promoters which are present. One is on PL, another one is PR, then there is PRM. Before understanding this particular diagram, we must have some more basic information about the lambda phage. For the sake of simplicity, I'm just going to start from the book Watson. Okay, I've told you what is a lambda phage, how does it makes a prophage. See, a lysogen is extremely stable under normal condition, but the phage dormant within it is called as prophage. See, in case if the DNA of the cell is being threatened with UV radiation, okay, in case the DNA has been damaged, thus threaten the host cell continued existence. This switch from the lysogenic to lytic growth takes place, and this is referred to as lysogenic interconversion, lysogenic induction. See, infection is going to take place. Once the infection takes place, then the lambda genome will be there and the lambda repressors will be there. See, this is the lambda repressor which is present. Now, if the induction takes place in the presence of UV light, this is how the cleavage occurs and this is how integration will take place. Now, before moving forward, you must understand about the genetic constituent of the lambda phage. Now, you would be wondering why we are talking about lambda phage, because there are two questions in CSR which are fixed from the lambda phage, and those two questions are not impossible to solve. They can be solved but require your concentration. So please do concentrate on a segment of genes present in the lambda phage so that I can show you which are the genes which are very important. You must know that the most important gene in the entire genome of the lambda phage is the CI gene, which is present, which is here, shown here. This is the most important gene. This is the gene that is responsible for the formation of lambda repressor. It is the one that will lead to the formation of the lambda repressor, and the lambda repressor once produced will lead to lysogenic cycle. It is the gene that will lead to the formation of lambda repressor. And lambda repressor will be the one that will induce lysogenic cycle. Okay. Now, please to listen here very carefully. The genes which are present are C3 and C1 CRO C2, C3 and C1 CRO C2. And then there are promoters which are present on the right hand side of the CI gene, is called as promoter on the right hand side. That is also referred to as promoter for repressor formation, promoter for repressor maintenance, Then there is a promoter just before the crow side, that is referred to as PRE, promoter for repressor establishment. Okay. There is a promoter that is present at the left side of the gene CI, that is referred to as promoter on the left hand side, PL. Now, in this particular sequence of the lambda phage, these 5 genes are the most important. Now, some characteristic features about the lambda phage genome. Some characteristic features. Which are the characteristic features? Features which are present. First feature that is present in the case of lambda phage is that it has both circular and linear DNA, both circular as well as linear genome, as well as a linear genome, a linear genome in itself. In itself. So when it has a circular genome, when it has a linear genome, that has to be decided. Now, first, linear genome is present in the case of lambda phage capsid. Linear genome is present in the case of lambda phage capsid. Lambda phage capsid. And circular DNA is present in the case of the bacteria. Circular genome. Circular genome is present in the case of bacteria. This is important. In the case of bacteria, the lambda phage undergoes circularization. Outside the bacteria, it remains within the capsid as a linear DNA. Because the size of the capsid is limited, as the size of the capsid is limited, so it will remain itself in the form of a linear dsDNA molecule. In what form? In this particular form. See, this is how it is going to remain. So it is going to be remaining in this particular manner, so that the lambda phage genomic DNA is arranged. This is how lambda phage genomic DNA is there. And lambda phage genomic DNA will remain in this particular manner. Okay. But when you look at the circular genome, in the case of bacterial cell, when you are going to talk about the bacterial cell, this is the bacterial cell. I'm just taking a bacterial cell for the sake of simplicity. Sorry. Okay. Now I've taken a bacterial cell. And in the case of the bacterial cell, this genomic DNA will get converted into a circular one. Okay. This will get converted into a double-stranded circular DNA molecule. The lambda phage genomic DNA is going to be integrated. It will form the dsDNA, circular dsDNA. Now, why does the circularization takes place? The circularization takes place because the lambda phage genomic DNA is fully methylated except at certain sites. Those sites are called as cos sites. Now, cos sites are the sites where the methylation is not there. As there is no methylation over the cos site, so cos site will be subjected to the restriction digestion by the bacterial restriction endonucleases. Cleaved by bacterial restriction endonucleases. Cleaved by bacterial restriction endonucleases. When they will be cleaved by bacterial restriction endonucleases, then the phage DNA will undergo circularization. This is how it will go undergo circularization. Now, moving forward, just clearing all the drawings. Now, in order to understand the entire mechanism of action of this lambda phage, we must concentrate over a segment of the genome of the lambda phage. Lambda phage has 50 kilobase pair of its genome with some approximate 50 genes. But to understand the entire process, we must focus our attention over the following 5 different segments of genes. Which are the 5 different segments of genes where we must focus? One is referred to as PL, another one is referred to as CI, PRM, PR, and the CRO genes. Okay, these are the genes on which we will have to focus. So you must remember all these genes and their locations. Now, how does transcription takes place? The process of transcription in the case of lambda phage or in the case of lambda operon is being regulated by 3 different promoters. The orientation of transcription is different in all the 3 different promoters. See, PR works away from the CI gene, which is the most important gene, and will lead to the formation of the lambda repressor. Okay, and will lead to the formation of the lambda repressor. Once the lambda repressor will be produced, then lambda repressor will lead to lysogenic cycle. It will catalyze lysogenic cycle. Okay. So what happens here is that the CI gene is being transcribed by PRM in one direction towards CI, whereas the PR and PL, being strongest promoters, will always catalyze the transcription away from the CI gene, away from CI. Transcription away from CI. Transcription of all genes, of all genes except CI. CI gene. Please, student, have some patience because this topic is really important and is one of the most typical topic in molecular biology. So you must give me at least 2 classes in order to make you even the basics understandable. Transcription of all genes except CI. Okay. So every gene is going to be transcribed except the CI gene. Moving forward. Now, the CI gene, this gene will be encoding a repressor, and this would be the lambda repressor. The CI gene is the one that encodes for a repressor molecule, and that repressor is commonly referred to as the lambda repressor molecule. Now, this lambda repressor molecule that is present consists of 2 different domains which are present. One is the C-terminal domain, and another one is referred to as the N-terminal domain. Okay, these are the 2 domains which are present. Actually, this is present in the form of an helical protein, and the 2 domains which are present here, they are being joined together with each other with the help of dimerization component. This is referred to as dimerization component. The structure of this lambda repressor resembles that of a dumbbell shaped structure. This is dumbbell shaped. It will contain the C-terminal domain, which will have the dimerization component, the tetramerization component. There is another region that is present that is referred to as the activation region, from where the activation of the process of linking of these lambda repressor molecules is going to be occur. Now, how does lambda repressor molecule controls the transcription? That we must understand. See, now, see, this is CI gene, this is the PRM, and this is PR. See, this is this particular part, CI gene, PRM, and PR. And then we will have a greater look at PL also. Now, if you look at all these diagrams, then CI gene has 2 different promoters, 3 different operators, and 2 different promoters present on its right hand side. On the right hand side, the first one is promoter for repressor maintenance. Another one is promoter for repressor formation. Okay, PR. Now, there are consensus sequences which are present within this particular sequence. There is another gene that is referred to as CRO, just on the right hand side of the CI gene. Now, there are two strong promoters which are present across this entire sequence, and those two promoters are referred to as PR and PL. Now, see this. These are the two promoters which are present. One is PR and another one is PL. PR and PL are strong promoters. Now I'm just going to tell you how does the process of transcription takes place and in what manner the transcription occurs. Okay, so please look here. PR and PL both are strong promoters. Okay, this is strong promoter and this is also strong promoter. When both these are strong promoters, these will catalyze the process of transcription to take place. One into the right hand side and another into the left hand side. Okay. When they will catalyze the transcription to take place, the genes will be transcribed. Now, once the transcription would have taken place, then the transcription of another gene will begin. One is referred to as the C-second transcription activator that you can find here. Crow gene. See, PR is going to transcribe the genes in this particular direction. PL is going to transcribe the genes in this particular direction. These are the two directions in which the genes will be transcribed. Then PR will transcribe a gene that is referred to as crow, as well as a gene that would be referred to as the C-second transcription activator gene. Okay, this is how. Firstly, the transcription of the crow gene will take place, and from here, the transcription of N gene will take place. So, which are the proteins that are going to be produced from the crow gene transcription? The crow protein. Crow protein will be produced from the N gene anti-terminus protein. Anti-terminus protein. Minus protein will be produced. And then there is PRE. Okay, this is also a strong promoter, which is referred to as promoter for repressor establishment. PRE promoter. Just a second. See, PRE. PRE is written as promoter for repressor establishment. PRE. Promoter for repressor establishment. Now see, which are the proteins that are going to be produced? Firstly, crow protein will be produced. Here, anti-terminus protein has been produced. After the crow protein, the second protein that would be produced is referred to as C-second transcription activator. Transcription activator. Now, C-second transcription activator, as the name indicates, it is a transcription activator. So it will be catalyzing transcription. Catalyzes transcription from PRE. Catalyze transcription from PRE. Transcription from PRE. From PRE. This is first level of regulation that I'm talking about. First level of regulation. In the first level of regulation that we are going to talk about here is that how does the first level of regulation is going to be influenced. In the first level of regulation, you can find the C-second is going to form the catalyzed transcription of the PRE that is referred to as promoter for repressor establishment. As the promoter for repressor establishment is going to be produced. So this is going to establish a promoter molecule for the repressor molecule. Now, when it is going to establish this particular molecule, it will perform transcription of the CI gene. Transcription of CI gene. of CI gene, and CI gene will lead to the formation of lysogenic cycle. Lysogenic cycle. How? I'm just going to explain it to you. And crow protein will lead to the formation of lytic cycle. Lytic cycle. So what is C second transcription activator going to do? It is going to C second transcription activator will be produced and will start the transcription for P from PRE. Okay. But this C second transcription activator is also regulated by another protein that is referred to as the host bacterial HFLB gene, HFLB gene, FTSH protein, host FTSH protein, FTSH protein. FTSH protein has an ability to inhibit and degrade the C second transcription activator. Catalyzes transcription of PRE, it is being inhibited by FTSH protein. FTSH protein of the host. As it will inhibit, the FTSH protein of the host is going to be inhibited. This FTSH protein of the host means bacterial cell is the product of HFLB gene, LB gene. And this HFLB gene rate of transcription will be high, will form high FTSH protein if environmental conditions are good. If external environmental conditions are good. Environment conditions are good. Please concentrate here. Are good. See, if the environmental conditions are good, the bacteria will be dividing. And for its growth, it needs to perform the transcription of HFLB gene. It will lead to the formation of FTSH protein. If conditions are favorable, HFLB gene transcription is high, FTSH protein is present in high amount. It will inhibit the C second transcription activator because the transcription activator will be cleaved by this particular protein. This will cleave. Once the transcription activator will be cleaved, it won't positively regulate the transcription of CI gene, won't positively regulate the lysogenic cycle, and thus crow protein, which was being produced, will establish the lytic cycle. In the case when FTSH protein is very low, lysogenic cycle will take place. Now, sir, why does such regulation takes place? Please, students, look here. Phage is very intelligent. Phage has made the transcription of its genes in such a manner that it could decode what is the type of environmental condition present outside the bacterial cell by sitting within the bacterial cell. Within the bacterial cell, phages are not having eyes to look when to perform lytic or lysogenic cycle. When the bacteria perform high rate of transcription, the phage has developed a mechanism in order to understand, oh my dear, bacteria is doing high rate of transcription. That means the external environmental conditions are good. So what should phage do? Phage should perform lytic cycle. Why lytic cycle? Because external environmental conditions are good. This means that outside there would be more bacterial cells to cleave. So the phage, looking at the protein of the bacteria, FTSH protein, decides that yes, it has to produce the crow protein necessary for lytic cycle and inhibits the transcription of the CI gene. See, if the phage decide that, oh my dear, FTSH protein is low, that means the bacteria is not able to synthesize the HFLB gene in adequate amount. This means the external environmental conditions are not good. Then the transcription of the C second gene will take place. It will help the establishment of the PRE, and PRE will lead to the transcription of CI gene catalyzing lysogenic cycle. If environmental conditions are bad, you perform lysogenic cycle. If environmental conditions are good, you perform lytic cycle. Any doubt if you have, you just ask it here only. See, bacteriophages, just by sitting inside the bacterial cell, decides if they have to perform the lytic cycle or they have to perform the lysogenic cycle. And thus they regulate their own rate of transcription by performing lytic as well as lysogenic cycle. Okay. Any doubt? Any doubt? Any query? This is the first mechanism. In the next mechanism, what we would be looking forward is how, sir, the lysogenic cycle and lytic cycle are going to take place. Okay. So this was the first mechanism of regulation that was taking place in the case of Lambda Phage. Now, what about the second level of regulation? Sir, the students might be queried about, might have a query that, sir, how the CRO protein carries out the lytic cycle and how the lambda repressor carries out lysogenic cycle. For the sake of simplicity, we must know that this CRO protein has an ability to inhibit all the transcription of the genes right from the PL as well as the PR. Okay, CRO protein will go and will inhibit transcription from PL and PR. Okay, as it will inhibit, go and inhibit the transcription from PL and PR. So which transcription is going to take place? The CRO protein will then catalyze the transcription only through the PRM that will inhibit the transcription of the CI gene. The PR and PL will be activated and all other genes will be transcribed. See this. Just a second. Just going to introduce that particular sign here. Okay, this is important. So scroll protein will bind at this particular position. Once it will bind at this particular position, the CRO protein will move from here. See, sorry. If this being the symbol of CRO protein, then the CRO protein is going to move from this particular direction and will bind at this particular direction. Will be inhibiting the protein, will be inhibiting PRM. It will go and inhibit PRM. This particular molecule will be inhibited. Once this particular molecule will be inhibited, then what is going to happen? Then no transcription of CI gene will take place. No lambda repressor will be produced. The transcription of all the other genes will take place from the PL as well as PR. Transcription of all the genes will take place from PL and PR except the genes of the CI from the PRM. So no other gene is going to be transcribed. All other genes are going to be transcribed. This is how the transcription takes place. Hopefully this would be clear to you all. In the case the lambda phage wins, in the case the CI gene wins, it will turn off PR and PL. See, if the CI gene is being transcribed, then what will happen? CI gene transcription will lead to the production of lambda phage. Lambda phage. Once the lambda phage will be produced, it will turn off transcription. Turn off transcription. The transcription. It will turn off the transcription from PR and PL. Okay. So this is how transcription is being turned off. Hope you have enjoyed the first lecture of the series of lambda phage. Okay. Please do have a look at it. Please do revise it. I do strongly recommend the book Watson, the book Prescott for having the same informations in them. Now, if you look here, these are the genes, CA genes, PL moving on the on the left hand side, PR moving on the right hand side, promoter for repressor maintenance, promoter for repressor establishment. And this is how all the genes are being transcribed. Okay. Now, early transcription genes, which are there. I have already told you which are the early transcription genes. the PL, the PR, and the PR, PL, PRE are the early transcription genes that are going to be transcribed. Okay. Then there would be immediate transcription genes, and then there would be late transcription genes. In tomorrow's class, we would be talking about immediate transcription genes and then late transcription genes. Hope you would have enjoyed the lecture. We will begin our class at 8 p.m. on the same timing. Okay, the recordings of the lecture will be made available to you. That's all for today lecture. Thank you. Thanks a lot. Good night. Bye bye.